What's up, everybody? It's Figure Friday, and today we have the sarcomere model. And the sarcomere is the functional unit of a muscle. So if you follow along and you watch Tissue Tuesday, we went over skeletal muscle, which showed alternating patterns of dark regions and then light regions. And I said that that was because of the protein organization in this sarcomere, right? So the sarcomere is region from Z disc over to Z disc, and it consists of actin and myosin. So you have the I band up here, right? That's going to be your light region. And then you have where the myosin filament is, the thick filament, you have the A band. So this is going to be a dark region, and then just a little bit or lighter region in here known as the H zone, and then the middle known as the M line. So this is when a sarcomere is relaxed. So what's going to happen is the myosin will grab the actin filament and pull on it in this direction, and we can see that that gap in the middle, that H zone, where there wasn't any sort of thin filament or actin filament, it decreases. And when we do so, the actual distance of the sarcomere, or the length of the sarcomere, shortens. And that shortening of the sarcomere is what causes the contraction. So this is what it looks like when the sarcomere is relaxed, and you can see there's not a whole lot of overlap between the actin and the myosin, the red and the blue. And then here, when it's contracted, look, we've got a lot of overlap between that myosin, blue, and the actin, red. And so there's an increase in overlap, and that shortens that H zone in the middle, and it also gets rid of the region where there's only the thin filament, that I band. So your I band shortens, your H zone shortens, but the A band stays the same length because it's the length of the entire thick filament, and that doesn't change. Now you know. Later.